Good afternoon, everybody, and welcome to the final session. And it's a real privilege for me to introduce our speaker, Professor Rob Bristow. I know a number of you will know Rob, but it's great to have him here with us this afternoon. Rob is Director of the Manchester Cancer Research Centre, has been since August 2017. The Manchester Cancer Research Centre, MCRC, is a joint partnership between Cancer Research UK, the University of Manchester, and the Christie NHS Foundation Trust. And part of Rob's remit when he came in to take over directorship of MCRC was to really bring with it and to pioneer a team science approach to cancer research across Greater Manchester. And that's what Rob's going to be telling us a bit about now. So, Rob, over to you. Thank you.
Great. Well, it's a real pleasure to be here today, and I just had a chance to whip around the rooms with all the different posters, but also the different kiosks, and it's really remarkable, I think, when Translation Manchester looks at its portfolio, and again, being this umbrella organization that really can bring people together in kind of a one-stop shopping where you finally have a lock that the key can go in, and then actually things are opened up. And this is really the reason I bring that up. It's very much what the MCRC is for cancer research in Manchester. So I'm going to talk about team science and translation with our particular approach. And I first wanted to start off with the most important slide, which is that whatever we do, it's patient-facing. And that means that the basic discovery research should touch the clinic in a translational pathway. And sometimes the patients are the ones that ask the most compelling questions, and actually sometimes the hardest to answer. And you can go through and any one of these questions that a loved one has potentially asked, or you yourself have asked, around risk, etc. And then people get excited about technologies. How do I get protons versus photons? But then something very simple: should I stop my metformin or statins? Something that is due to another comorbidity during treatment. And these latter questions we actually don't have any answers for, but I'm going to argue through team science and the approaches that are happening in Manchester now that actually we can really start to have clinical insight from our data and feed back into these questions. So the other piece I think that was about today, and I was watching some of the sessions, which is really about innovation and that creativity. And how do you instill not only an ethos of commercialization and innovation with the university, but actually allow for creativity, allow for brain time, allow for people to come into a room like this and for 2 hours think of everything and anything they could do, potentially with a carrot, not a stick, hopefully these days. And to think about how to apply research so that actually it winds up either as an assay, for example, in a laboratory, changes the way we think about things, or it is on the benchtop of an NHS facility and it's actually giving information regarding patient care. And so, innovation is absolutely a prerequisite of commercialization, because you can't commercialize something unless, again, it's added value to what already exists. So the MCRC then is committed to this principle of synergies in soil, and I'll come back to it. I used to just say soil, now I say synergies in soil, for innovation, and we use team science to drive that. So let's explore a few areas together. It's the concept of the MCRC, team science defined, and maybe some exemplars. How do you build new teams? And then also, what about environments where those teams sit? And of course, this is a very exciting time for cancer, given the new Patterson rebuild that is down on the Christie campus, where there's a co-location of both researchers and clinicians. And I have no disclosures for this talk. So let's talk about the concept of the MCRC and team science and how we pull it under one roof. And again, as I said, Translation Manchester today is doing the similar thing. So as Nigel said, on the right, the Manchester Cancer Research Centre is really a synthetic body that takes, again, the very best of what we have in Manchester and puts it into an umbrella cancer centre that allows us to lever the partners back and forth, and then also allows us, of course, importantly, to have outputs and impact as a cancer centre. So we start off with the university, where, of course, we have a beacon in cancer, Cancer Research UK, where we have multiple infrastructure awards, as well as a basic and discovery institute, and the Christie Hospital, at least as a founding partner with those two other institutions in 2006 for the MCRC, which is Europe's largest single-site cancer center. And what comes out of that, of course, on the far right, hopefully, is really impactful grants, impactful science that makes it to the clinic. But we do have to think about the Greater Manchester Cancer Plan or those NHS long-term goals for early detection as well as for precision medicine. And of course, a number of these are going to be activated through our academic health science plan and commercialized potentially through Health Innovation Manchester. So I think what we've done in the last 5 years is we've started with the partnership where Nick Jones left it. And now I think we've broken down silos and we have very strong affiliations for the MCRC with Manchester Foundation Trust, Salford Royal Northern Carolines, and then also are enacting different projects associated again with these other partners down at the bottom. And the other piece, again, is that we've designed this to be almost like a National Cancer Institute in the U.S., comprehensive cancer center, because from the outside, in terms of international competitiveness, you want to be like something that somebody already knows about. But inside, you want to have specific USPs. And importantly, I think this is true for the BRC. It was certainly true for the Cancer Center application that we submitted before the BRC, which is really a focus on diversity and multimorbidity and how those two, again, come together to inform on cancer patients. And I think about, Nigel, what was it, 3 years ago, 4 years ago, we started talking about this and having workshops, et cetera. And this really stimulated us in cancer to think about. So a very large portfolio of research, more than 240 million in active research grants, lots of samples being collected for translational research, and lots of cancer researchers. And then again, important institutes or centers that are under the MCRC umbrella, and that goes all the way from the Manchester Cancer, the Manchester Institute in terms of basic and discovery science, right through to, for example, INCITA and other infrastructure awards for imaging, radiotherapy, etc. And so when we think about the Translation Manchester footprint on top of the MCRC, certainly we have aspects that are associated very much with, again, the early the early discovery science piece. And then in the NHS, of course, by definition, again, with a large cancer hospital and one of Europe's largest phase 1 trial units, driven through the CRF that is funded by the NIHR grant to MFT and Christie, we have really that middle part now, which is the translation, what we've been talking about today. And in selected aspects of these, the MCRC shamelessly steals in order to have really effective partnership that includes with the Pankhurst Institute, with the Innovation Factory, and of course, as I said before, the CRF and the Health Innovation Manchester. And altogether, then, that forms a milieu in which we can drive personalized cancer medicine in Manchester at an internationally renowned level. So we've recently had a series of experimental cancer medicine center competitions, of which there are 8 in the United Kingdom. We certainly have ranked very well in that competition. Official results haven't come out, but unofficial results have. And it's because, again, a team science associated with the early phase investigators in the Christie aligned again with very strong biomarker programs, for example, the National Biomarker Center with Caroline Dive. But you can start anywhere in this circle in terms of the interactions, of course, but the Cancer Center really is sitting in the middle of this, trying to, again, facilitate different aspects of it. So in terms of the Cancer Research UK and, around the 2 o'clock position, really, again, it's about that infrastructure that we have, protons versus photons, biomarkers, all those good USPs. And how do we drive that then into the ecosystem to have personalized medicine? Of course, you have to have the university in not only basic discovery research, but also line management. So there has to be a career piece, both early careers and late careers. And everyone has to see their success through the Cancer Center. Importantly, governance around specific gene sequencing or the impactful nature of what is happening at St. Mary's in collaboration with the Christie to make sure, again, that NGS and other targeted DNA mutation are being annotated and placed in the chart. The sample collection, again, for patients that come through in the MCRC Biobank. And then, of course, this large hospital that actually, if you can have all of that data in a digitized format and can look at real-world outcome as well as electronic patient-reported outcomes, this becomes a very exciting area for research. But also, there's been some really strong, again, pushes of national clinical trials from some of the translational research that started in Manchester. And one is Target National, which is using liquid biopsies to drive personalized medicine. So the use of ctDNA, which was started actually by a grant from the Christie to Caroline Dive and Matt Krebs. And that turned into a Nature Medicine paper. And now it's a 6,000 patient across the UK study. The other important thing, and of course, is commercialization. I think it's fair to say when I came, and you know, the last few years or so, it's been very sobering to see how poorly we're doing in commercialization innovation in cancer in Manchester. And that's not because there aren't good ideas. Again, I would argue that when I came, there was a culture in which commercialization innovation was an afterthought as opposed to a first thought. And how do you set up cohorts, for example, of trainees and clinicians and scientists to think about that conduit. So we've really tried to step change this with a collaboration with CRUK as well as the Innovation Factory, funding a new commercialization and innovation officer, Natalie Dolan, who is surveying all of the potential assets that we have within the MCRC for commercialization and innovation, and also trying to help again with bilateral, tripartite types of complex contracts when you have a trust in a company and again the university. And for reasons that I'll talk a little bit in a moment, we focused on digital assets and advanced materials as those areas that we think we could easily commercialize in the near future. And just in the pink bar, you can see where Manchester ranks across the number of CRUK centers and institutes. So not really bad, but not really good. And of course, we want to go to a level that is in that CD area at least in the next 2 to 3 years as one of the endpoints to this. So, you know, coming to Manchester was really interesting because you're wowed by all the technology and the hospital, etc. And I heard about this devolved healthcare system, but didn't realize, coming from Toronto, the really diverse population that we sit in. And at first, it was a bit of a challenge to think about how are we going to actually drive our programming into communities that instead of hard to reach, we call it now time to reach communities. And that could be due to differential ethnicity, socioeconomic factors, multiple comorbidities, etc. But this became an opportunity for research, of course, because when you look at the map of which cancer care is given within Manchester, those one-year survival rates, actually they're slightly improved now, but the one-year survival rates were all under the national average, which was about 72, 73% when I came. It's now 74%. We have some, I'll just say, higher socioeconomic regions that are actually doing better than that, but we have many that are doing less than that. So these are one-year survival rates, not 5-year, one-year survival rates, of which some localities have, for example, 57% versus 75% average. So that's extraordinary differential. There may be multiple reasons behind it, but one of the things that we're thinking about is, is this due to the comorbidities? Is this due to engagement? And how do we really put in interventions to change those numbers around? So the first thing that is happening is in collaboration with the Greater Manchester Cancer Plan, there's a tableau, an actual approach in which those constituencies now, due to postal codes, are given one-year survival rates and stage of presentations, so we understand those late-stage presentations relative to early-stage presentations, as a way to understand what the delta would be to put an intervention into these areas to improve the results at the very, very top. And those interventions, again, are based on a number of pieces that I've already described in the second point here, and that includes also mental health as well as physical health and engagement. And there's another piece here, which is if we can get patients engaged and they're coming to the Christie, for example, for precision medicine approaches, that usually means that they maintain that engagement. So, for example, we have electronic patient-reported outcomes of which the Trust put £4 million so that all patients at the Christie would be able to enter their side effects, both from a side effect standpoint as well as their experience using a smartphone. And what we found is that even if patients came from, again, socioeconomic deprived areas, once you were in the system, they kept on being in the system. So it's not like they weren't keen to not be involved, but actually you could start to think about a hospital in the home approach for these patients and really taking things to them where they're engaging actually through their smartphone. And I guess the other question that we have, and we started some studies on this, is that if you have cancer and a comorbidity, what does that mean for the cancer biology? If you have diabetes mellitus, type 1 or type 2, and you have cancer, what's the metabolism of the host and the cancer doing in that particular state? And so if you have good clinical data, you can start to parse that out a little bit. And then, of course, we know now in the United Kingdom, there are different risk factors for different ethnicities. And, of course, we heard different mortality statistics for different ethnicities during COVID. But I think really now we're really focusing back on ethnicity. I'll finish off really speaking to some of our approaches in that regard in terms of social responsibility in a moment. So our vision is cancer precision for all. This is what we put into our cancer center, our CRUK center grant, and you can read again the italics. But importantly, the points are that we have medically and socially complex patients in our area, and some of them are excluded from these great precision trials that we talk about, where again we use a liquid biopsy to match to the brand new treatment that's coming from our industrial pharma partners. And we think that the patient's underlying health, those comorbidities, are affecting their cancer outcomes, either side effects or potential response to treatment. And I would hazard a guess, for some patients, it might actually improve outcomes. So we know, for example, metformin in preclinical studies actually decreases tumor hypoxia by changing the mitochondrial axis. And in fact, that's been shown to improve results in retrospective series, and can we actually see in a prospective trial that that might happen? The other piece is around, again, ethnic diversity. And there's increasing data that suggests, again, that ethnicity is associated with differential genomics. And again, how well are we doing in terms of capturing that information? And then, so the complexity and diversity of the vision defined by the MCRC absolutely requires team science in order to address this. No one investigator can actually look at this complexity by themselves. And the other piece, again, afforded again as a promissory for our region, is to have a digitized healthcare record that you can search in real time. So at the Christie, in addition to the electronic patient-reported outcome, we're also activating a real-world outcome with our bespoke electronic healthcare record. That means within 12 hours or so, the record is live. So we can capture the real-world data, and that includes both the data of the Christie, or now because of the integrated care record at GPs and other trusts, we can analyze that. And we can put in context, again, using AI-driven clinical supports with the Pankhurst about what's the best route to go forward in terms of using that information to think about new ways to treat patients. And then we have to implement that, and that's a practical approach. But of course, it also is important to understand the inclusive piece to that implementation and also the impact. And this rapid learning approach is a different way of doing clinical science versus the kind of randomized phase 3 trials, in which this rapid learning approach can actually have sequential cohorts and you can change treatment in the NHS with it. We also have a patient data promise, so we sat down with our patients and said, what can we do and what can't we do? So we actually have permission from our surrogates in our focus groups to actually use that comorbidity data and actually to potentially interact with pharma or companies that actually can look and make sense of the data in order to improve cancer outcomes. And then an exciting piece that I think is that we're going to electronically link the biobanks, all those specimens, directly to a patient's outcome. So a scientist now who's interested in translation can come to the biobank and ask for responders and non-responders and get samples out and actually do a test or an assay, which is very different than currently. You have to really, you really don't know what are the responders and the non-responders associated with those biobank samples. So I think we'd be one of the first in the world if we do that. And then, of course, also the important thing is how do we think about who's leading our programs when we think of team science. And I think we're very fortunate to have, again, a university that is activated towards social responsibility on the far right, and individuals who sit on a number of research programs, let alone the MCRC steering board. And then also on the far left, again, international collaborators who are world-renowned for the research they've done and are driving, again, some of our leadership in Kenya. And again, the MCRC is very much takes to heart the diversity within our populations, and the center has an ongoing lecture series with the Barts Cancer Center in London to really explore all of these different areas and how, can we make sure that we're inclusive when we think about our engagement, but also, really importantly, how we think about our interventions in research. Okay, so that's the MCRC. So we're very excited about the portfolio we have. But at the same time, you have to focus down and you have to prioritize because there's not all the money in the world. Certainly we know for the pandemic in terms of financial contracture. So what are some of the intramural and extramural exemplars about team science at the MCRC? And also, what were these town halls that we did and how does that concept kind of go forward? So the town hall concept when I came was something that we used in Toronto, which was to bring people together, kind of lock the doors, figuratively, not literally, and actually, again, say to people, this is your brain time, all right? We're going to dangle some money in front of you, and we're going to ask you to do a few things. The first is to bring a new idea to the forum and to think about what would you like to see as a headline in the Times or the Guardian 3 years that spoke to, again, a Manchester ambition and potentially a Mancunian project of where that could only be done in Manchester. And how do you interact in that forum when we put scientists and clinicians together, but most importantly, also put patients in the same room? because you have a very different atmosphere when you have patients in the room in addition to scientists or clinicians, I can guarantee you. And the second is that what this drives, again, and we did this in a tumor site-specific way, breast cancer, prostate cancer, really for practical purposes, but we had physicists, we had chemists, we had clinicians, and it was really to drive goal alignment across the team. Could we get to a headline? And actually what we did in the last 10 minutes was actually said what the headline was. And what was really interesting is for two of them, we actually had a reporter from The Guardian that was shadowing and actually wrote this up for an article and said that if there's one thing that she knows, is scientists and clinicians are terrible at having actually headlines in the lay press. But everybody tried, and I think that was really, really important. And the last one is really important too: a high diversity of membership. And the best ideas will come from diversity. It's just always the case. So why did we do this? Well, it started for me actually almost 10 years ago in Toronto, because we were given an extraordinary amount of money in Toronto, so $20 million to sequence prostate cancer during the whole genome sequencing era. And this was very exciting, and we had to deliver 500 genomes. And so the main project, as it turned out, was that we wanted to drive all of that science towards understanding who had hidden metastases and who did not at the time of surgery and radiotherapy. And we got a lot of really high power, and we still have a lot of high power manuscripts that have come out of it. But importantly, that group continues even after the ICGC project was over in 2018 as a pan-prostate group, and we have over 1,500 genomes that we can ask questions around, all harmonized to the same bioinformatic pipeline. So setting up a team of 70 people then who had never met each other, where you had sequencers, bioinformaticians, clinicians, and then basic scientists was really an exciting piece. Lots of roadblocks along the way, but we hadn't done any genomes in 2011. By 2018, we were producing at the highest level. There were a number of things we learned from that. The first is that because it was a contract, we had to get through 500 genomes. It meant that the main project and the main question, again, with goal alignment, was always going forward. But early career investigators could get, as the data came through, could ask questions around some of that some of that aligned data. So we actually had 4 new careers driven by this project in circular RNA, proteogenomics, enhancers and silencers, and microRNA, that took data that was coming from the main project and actually aligned it to, again, putting in program grants and getting their grants. So I'm very proud of that particular aspect of team science, where it may start with senior investigators for political reasons to get the grant, et cetera. But again, there has to be room at the end for those early career investigators to take their slice of the pie. So what did I learn from this? Strict outputs are really important. Monthly meetings, making sure you're on target. You have to change your language so that you're understood by people from different types of backgrounds. and it was always very interesting for a bioinformatician to talk to a clinician and vice versa, and realize after 5 minutes they had no idea what the other person was saying, and you had to really think about changing your language because of that. The troubleshooting can be shared across disciplines. That's actually really exciting when you're stuck that someone else can have a good idea. We kept to tight guidelines because you use a scientific advisory board. And also importantly, we shared first and senior authorships up front. So actually there were junior career investigators who got a senior authorship because we might have 5 seniors and we might have had 3 or 4 first authors. And that's because it was such a big project that actually there was equipoise in the amount of work that was done in the different units. And we decided that up front. The other important thing was to keep the politics within the senior people. Junior people don't need to be bothered by that. That just slows them down. So the senior people actually did a kind of a pinky agreement that we would actually keep the politics away from the juniors and solve that. And I think that helped those 4 careers very well. So what does this slide mean after that slide? This is what I'm doing now, okay? So that was a great stepping stone. And coming to Manchester in the last 5 years, we've built new teams now around spatial multiomics and spatial bioinformatics, and great collaborations with Magnus Rattray and David Wedge in the bioinformatics side, with the core facility in the university and also in the MI. And I won't go through all of this, but it just shows you again that when we set up these teams, these translational teams, what we can do now as we embrace the new technology and now ask really exquisite questions, not on the whole genome and bulk sequencing, but actually on the intraprostatic architecture, and where did these clones come from that then spread to other areas of the body? And can we develop a biopsy team at the Christie? The answer is yes, that we can go and biopsy those metastases before any treatment to understand the natural history of the disease. So a very exciting time to come to Manchester, and also another exciting time for transdisciplinary and transfaculty science. Again, the team has grown to include trying to create human chromosomes with the Manchester Institute of Biotechnology for new models in cancer. And on the right, thinking about how to work with engineers and chemists with companies that now make the latest in mass cytometry to actually look at protein-protein interactions and protein complexes. And this is all going, and it really started again from some of the ideas that we had, not in the site-specific research, but actually research just around prostate cancer and how far could we take our program. So very excited that these areas are being funded. But at the time, we basically had these town halls, and that is a picture of me driving one of them. So it wasn't a board meeting, it was a strategy meeting per se, and had no preset agenda. In fact, if anyone came in, they said, oh, I know what we're going to do, because here I have a great idea for this. It was just thrown out. I wasn't interested in it, because it had to be a collective approach. And again, we had juniors up to extremely senior people, both in terms of the scientists as well as the clinicians. And this was the speed date, right? In 2 hours, what was a new idea? And we said, we'll give you real-time outcome. We'll give you access to genomics and biobanking. We'll have a cancer economic piece. And then also you'll have access to trainees. And so what's your headline? And so some of the problems and challenges that initially there was resistance from principal investigators with this approach. So, why are we doing this, and why are we involving so many people rather than celebrating the individual, the superstar? And it's a different model, right? And people have to kind of trust the system. And if you haven't done it before, it's very hard to trust, because you might not understand what's the return of investment when you do this. As opposed to if you have one investigator, you can throw them against the wall and say, we gave you money, what did you do with it? If you've got 40 people all together, it's a little bit harder to stick, right? And the other important thing was to involve more than just one trust. So the Christie clearly is a big trust in cancer, but a lot of cancer research is ongoing, again, at Salford Royal as well as MFT. So it was trust independent, even though the monies that were given for this were actually coming from the Christie and from the university. And importantly, those patient advocates. We had two of our ideas that actually came from patients. I remember the lung cancer one very well. There was a lung cancer survivor in the front row who said, are Manchester cancer patients any different than anywhere else? And Caroline Dive was there, and Colin Lindsay, at the time a young medical oncologist, and I looked at them, I said, what's the answer? And Caroline Dive said, I don't know. And I said, but we should. And actually that started off to find out that there was a unique RAS mutation that for some reason is at Manchester about 50% incidence, and the rest of the United Kingdom is 10%. And there are lots of potential reasons for that, including populations not moving during the Industrial Revolution. But actually, it made a mouse model, and we actually have drugs against this now with our partners, and it gave Colin Lindsay a career in terms of RAS targeting. So it was a really exciting project, and I've already discussed again the remit that we had. So here are just some of those headlines on the far right. Young women with breast cancer reaching the unreachable. Manchester banned sunbeds. So the melanoma one was a social policy piece, of which we have had two recent publications suggesting, again, what would the economic benefit be in the healthcare system if we reduce melanoma rates by getting rid of sunbeds. Hem-onc was about actually bringing the three non-aligned at the time transplant units together to think about graft versus host disease, and this actually drove a career of Mark Williams, who's recently got an MRC Clinician Scientist Award and sits in the Manchester Institute, who was just really a baby clinician scientist when this started, and his idea was to use MS swath as well as mass cytometry to try to understand the immune cells that were happening in patients that had graft versus host disease. And then again, we talked about the lung cancer piece, and then more recently a hepatobiliary one where we looked at, again, the idea of inflammation in the liver, both with our non-cancer colleagues who are hepatologists in the university and at MFT, as well as our clinicians. So we had other tumor sites that were developed, again, with headlines from a different approach. But importantly, this return of investment was reported to both the Christie and the university. So we got a big ACE grant, so Alliance for Cancer Early Detection, from the breast cancer idea, new early career investigators, a new social policy, and also trust alignment through this approach. And also, and I won't go through this, we were able to then start to pair very quickly preclinical scientists with clinicians who had goal alignment, at least within a tumor site group. So if you said it was breast cancer, you knew who the preclinical researchers were, and you would know who the clinicians who were activated in translational research. Same thing for prostate cancer, same thing for lung cancer. And we were surprised sometimes because people who were in the room who had never done breast cancer research came because they were really interested in a specific question, and they got aligned. So I'm very proud of that piece where we do align on the tumor side and the host side with these different elements. So the benefits then is we got new collaborations and narratives and grant opportunities. And chance only favors a prepared mind. That was the story for the breast cancer town hall, because although £150,000 was given to drive the question about how to use polygenic risk scores from a cheek swab in women aged 40 to 50, and then bring them in for a digital mammography piece, that actually was being activated across the Greater Manchester Cancer Plan, and we used that actually as the basis for a grant of $3.3 million for the to become one of the 5 members of the Alliance for Cancer Early Detection, in addition to our UK counterparts, Cambridge and UCL. So we gave them a different version, but it all started actually with that particular town hall. And again, you have to have pump priming funds, either in kind or actual funds for this. You just have to recognize that the different partners will see different ROI. So the university wants to see maybe PhDs. The trust might want to see a change in patient care, and not all of these ideas will give everything. But importantly, it drove research brands for the MCRC and really started to narrow us down. And also, those narratives are good for philanthropy and fundraising. And finally, it was a chance for early career investigators to shine and to take a project forward where potentially they wouldn't have had the opportunity before. So what did we get from 150,000? Well, we have this, the Alliance for Cancer Early Detection. You can see the institutes on the lower right. We've hosted a summer school. We have a number of foci in different areas. The original grant was $3.2 million, but now we have another $2.3 million in subsequent ACE funding. And I led a grant with Cambridge where we just received $5 million around detecting cancers with immune system approaches. So at 11 ACE PhD students. So for 150 grand, I'm going to argue this is good ROI. Similarly for Radnet, 18 million in funding that we have by bringing investigators who were doing radiotherapy with investigators who are interested in comorbidity in the university, in cardiology, in respirology, in brain injury. So we have a number of work packages, of which they're all transdisciplinary. They all involve new collaborations with some that were non-cancer researchers. We brought in David Wedge into work package 3. We got him from Oxford, and he's really driven a lot of exciting bioinformatics. And the impact and the promise of this are shown on the right. But actually what's really important, again, in terms of return of investment, is that we've levered a further 22 million in grants and have 19 PhD students associated with this grant that went in 2019. And this is just, again, now the mechanism meets machine vision of Radnet, where, again, in the terms of the translational circle here, and it's particularly inclusive with respect to complex patients and comorbidities, that in fact the new hypotheses and novel model generation at 3 o'clock is actually something in which we're taking animals, for example, that have atherosclerosis or have diabetes, etc., radiating proton versus photon and understanding the tissue injury when you have a comorbidity. And furthermore, there are novel models now looking at normal tissue toxicity and the immune system. So Doug Dyer is working with Tim Illage. I think there's a poster here at this meeting, again, to really look at mucosal immunobiology and how that's reflected in radiotherapy. So we've done extremely well with this transdisciplinary approach, and that it reflects back on some of the populations with the comorbidities that we have. And one of the things, of course, is proton therapy is very, very expensive. But if you used it in patients who had comorbidities to kind of come off normal structures and give a better chance of cure, but a reduced chance of side effects, then that's a new indication for proton therapy. And just very, very briefly, big programs in biomarkers, as I said already. Won't go through all of this, but again, the Biomarker Center is really a translational hub for us. And we pull the very, very best science, put it into the biomarker program, reflecting back on clinical trials. And we have resistance, then that can go back into the Biomarker Center. And again, we can look at liquid biopsies or even potentially proteomics of tears, as you'll hear about in a moment, to understand about both the risk of cancer as well as the response to cancer. And other areas that I'm really excited about are this integrative pathology piece, where the Biomarker Center will actually be right next door to the MeBe-TOF machine that I showed you before. We're developing novel mass cytometry approaches. The hypothesis is that more is expensive, but more is better in terms of getting the patient's precision medicine right. And so we have and now an approach with David Wedge and myself and others about how we think about tumors and evolution and developing evolutionary pathways quickly with AI approaches, and can we block evolution by that approach and understanding again what's happening within the cancer and then metastases from that cancer. And setting up a brand new program actually in multi-omic bioinformatics, and actually we'll have some teaching workshops that Manchester will lead for the UK in that space. And then liquid biopsies now in the radiotherapy realm. So using, again, an MR-Linac, where you have an MRI machine associated with a linear accelerator for treatment, and you can take multiple images and acquire multiple biopsies and actually think again about composite biomarkers that might come into play. But importantly, cross-training teams in medical physics, in biology, and again in radiomics, and feeding in again to our Radnet network. Couple more just exemplars. One is Team Brain Anatomy. Very proud of this. This is a pediatric oncology grant, Stand Up to Cancer, Cancer Research UK, only 3 in the world. And this was a collaboration with St. Jude's and Groningen to look again at proton therapy and neurocognition in children. It's driven by Martin McCabe and his team. And again, it really just puts Manchester on the map because of proton versus photon cranial radiotherapy, but again associated with a huge childhood radiotherapy center in North America and a preclinical program of rat proton irradiation in the Netherlands. So again, we should be very proud of these team science approaches at an international level. And the other thing about synergy in soil, we've done this a number of times. You know, CNS was not a strong site for us when I came. And the Jeffrey Jefferson Brain Research Center has really increased the brand around neuro-oncology, around stroke, of course, and dementia. And we had a number, again, of researchers who were interested in CNS, either Gerben Bors, who's a clinical academic, Kay Williams, of course, who's been interested in brain for a long time, Ben Dickey, in terms of neuroimaging, and then Marianne Asnar, a medical physicist. And this is just the Radnet piece, and you can look around of all of the different assets that we had. And we use this to entice a researcher to come to this soil and to synergize with this group. And so I'm really was happy on September 1st for us to say that we had recruited Petra Hammerlich, a hybrid investigator from AstraZeneca, and will be at University of Manchester looking at novel models, preclinical models of brain tumors, currently as a director of drug development in glioblastoma at AstraZeneca, and as the new brain tumor charity chair in translational neuro-oncology. And she came because she could see, again, how she would be able to catalyze across the different piece, and very excited about the team science approaches here. So again, Chance, absolutely favored the prepared mind here as we thought about the tumor microenvironment and how to build CNS, and then look for an individual who would be interested in the tumor microenvironment. And one of her assays is actually using tears to understand, again, early development of tumors within the brain. So what I'm really excited about is now that we had an international advisory board just 2 weeks ago where we showed our wares to them, and they said rank us internationally for what we were doing in this team science approach. And we got very good feedback, and that feedback went straight to our leadership of Nancy, Graham, Roger, and Ian Fowkes at CRUK. So our strategy now of those colored circles, and you can pick any one that you're interested in, is actually informed by the town halls. It's informed by the impact of the team science that we started 5 years ago, but also it's now informed by large infrastructure grants. And I wanted just to take a little bit of time in the last 4 minutes to talk about 2 things. One is the global genomes, which is team science international level, and 2, that building in the middle and how we think about team science with a new infrastructure. So in terms of international collaborations, lots of cancer centers can put maps out like this, and we do have a lot of collaborations, and there are lots of reasons to do international team science. That's because, of course, you get critical appraisal from your peers or different healthcare systems. Scientific innovation occurs in different countries, and you can do a synthetic approach to your own work. But importantly, especially when you work in lower and middle income countries, there's an altruistic approach, a humane approach, and frankly, it's the future for impactful work from our institution to show that we actually affect not only UK healthcare, but also international healthcare and international science builds. So I showed a map on the right before, and there's a glaring piece of information that is missing. You can see a lot of boxes are over the continent of Africa. Not one project in the ICGC came from Africa. So what does that mean now? It means that if someone comes in from African continent into the Christie, we don't know anything about their genomes other than what we might extrapolate from a Eurocentric patient. And this is how bad it is. So if you look at the human population according to the United Nations, and let's say Africans are somewhere in the order of 16 to 20%, and then go around again this translational pathway, and look at, again, at cell lines, models, and fundamental research, particularly genomics, of which there was an ICGC and TCGA, and this was supposed to be the world sequencing all the tumors. 91% are white people, and less than 2% are black people, and particularly in this case. So this is bad, okay? This is not precision medicine. And it's even worse when you start to think about the lack of models, right? Just keep on going down. So we have a chance then in the Cancer Center to do this differently. And it came through this particular interaction with the Kenyatta University Teaching Research and Referral Hospital, reciprocal visits from Kenyan cancer researchers and ourselves, and again, memoranda of understanding signed off with the UK Kenyan High Commission between the Christie and the university, and also this new cancer center being built at KUTRH. And we put in a grant to NIHR to say we're going to do early detection in actually a cancer that matters to Kenyans rather than a highly prevalent cancer in the United Kingdom. And that was esophageal squamous cell carcinoma, of which usually it's esophageal adenocarcinoma here in the United Kingdom. And we're going to do best engagement through 5G, because all of their phones have 5G in Kenya, ours don't. We're going to create a hub and spoke model where the data is uploaded and have a digital cancer center there. And we're going to do whole genome sequencing and tumor microenvironment assays to understand why some Kenyan counties have a higher incidence of this disease versus other. And the outputs in terms of sustained outputs are shown on the right: capacity building, novel science, approaches again in healthcare systems. And very, very excited about this particular grant that we have. And more exciting was that this is a co-created grant, a truly co-created grant where we shut up, finally when they told us to shut up, we shut up about saying what we were interested in, and we listened to what they were interested in, and then we developed the grant around them. And so to me, it was an exemplar of not only team science, but active listening to a partner that has a very different need than yours. And importantly, this is really, by Kenya, for Kenya, and the 3 million grant is now operationalized, and we'll get some really exciting science out of this. So in the full picture here, when I think about what we could have with our internationalization piece and working again, developing the genomics and the biology of tumors that are in patients who are not Eurocentric, then putting that data then into the sort of cancer intelligence piece about the multimorbidity, you really, I think, can change on the far right what precision medicine means. And so that's onus is on us for the next 5 years to show that these interventions, that the collection of data that actually will lead to a precision medicine approach, for example, a black patient coming in from Africa versus a Eurocentric patient, and that would be very exciting. More exciting would be someone at ASCO, one of our main meetings, actually presenting that data who's black and who comes from Kenya, rather than, again, a white person that represents them. Okay, so last minute, boy, wouldn't it be nice to move into a new building? So we had a chance through serendipity, right? Well, not serendipity, through actually a tragedy, of which just before I came, I had nothing to do with it. The building burnt down, the Patterson burnt down. And this was a really difficult thing because people had to be spaced out and have been spaced out down at Alderley Park. But this new building is almost done. It'll be completed physically by end of January of next year. People will move in probably around April, May. I won't go through all of these because these are really exciting pieces. But this is the first time that a CRUK institute is attached to a cancer center in the United Kingdom. And also, we're going to be really excited about how we tell our stories, our research stories, to the public. So the guiding principles here is again about team science, where patients are part of the team, and how we tell our stories and how we think about the collaborations. And so we've been having a series of synergy groups, really bringing the teams together, again, patients and scientists and clinicians, to say, how best do we do pathology? How best do we do radiology? And one of the things that our researchers wanted was, could we have on call, just like you have on call of patients, could we have on call to the research, a pathologist or an imaging expert, so I could take my section of a tumor, either human or for a PDX, or, for example, a genetically engineered mouse model, and actually have somebody and look at it for me anytime during the working day. Similarly, I might have an image, a PET image or an MRI image, again from a clinical trial in humans or from a preclinical. Is there somebody that I could go at any time during the day and actually get feedback on that model? And so that's actually the promise that we have from imaging as well as pathology in the new build, that there will be, again, people who will be staffed to work with researchers and to give real-time information on when they come in. And there are a number of other things I won't describe here, but these synergy groups are really about how best we bring out team science, how we drive, again, interactions between scientists and clinicians, and how we use the new building to do this, not only about who sits where, but also the collaborative spaces that we're building overall. And some of that is about telling stories. So for example, on different floors, when you get off, there'll be a specific image, again, with a QR code that you can basically look at and see the patient story behind that. And these can be changed, from time to time, just in terms of the way that they're applied. And for the clinical trials pieces, clearly, we're going to see, I think, imagery around those. And the important thing, of course, is that from the outside, whether you go in or even if you're walking by when you have digital screens, our stories also will be told out to the project, how we translate, what this building is about, why Manchester, what's our USPs. And I think we have really, really compelling narratives. So to end then, for team science and an inclusive MCRC, I think what we've put in play in the last 5 years have developed some great teams, have developed big money to come into Manchester because those teams are actually transdisciplinary and transfaculty, and will continue to drive, again, that translational piece into the clinic, and then also reverse translate. Again, we think about the host as well as the tumor, and the host here is also the complex patient, and try again glean clinical insight from that real world evidence piece that we have. Inclusivity is biology, right? It's not just fair, it's biology as well. And again, setting up teams to explore that and explain to it. Right now, if you're in the United States or anywhere, you can't actually run an ethnicity-specific trial, so the FDA will not allow it. And they want to see the diversity data that actually suggests that there will be a difference, for example, based on a different genomics. And that's something that we really could lead in that particular era. And then finally, in terms of complex cancer data associated with the Greater Manchester Integrated Care Record and all of the things that are going to happen in the Biomedical Research Centre, we've already started a collaboration with CRUK to be a collaborative choice within the United Kingdom and drive training in this area, but also novel clinical insights. So great day for Translation Manchester. You've seen this slide, really, really exciting. I just want to say our team science approach is very much similar. I'm really excited what might have come out of today and will come out of today, with these types of approaches. And so it's great to be a partner of Translation Manchester, but also it's great to be in Manchester. Thank you.
Thank you very much, Rob. Rob's agreed to take a couple of questions, so if anybody's got any particular question for Rob at this time, please feel free. Simon.
Yeah, so thanks for the keynote. It was really, really good, and that's not from a medical person. So,
Phew!
How, team science seems to be a big thing, and you seem to be a big advocate for it. So how do we get that team science into the university? How do we move the barriers from the university, especially where we might not have such strong advocates for it?
Yeah, well, so the first thing is we must have strong advocates. So, I think the university does talk about it, right? We have one person who talks about it a lot. I think, one of the difficulties is, and I was hoping it would come up as a question, is, how do you define someone's role in a team? And this is usually at a university level sometimes very difficult. So, as I said, when people are really cranking, thank you, just being told, go to the microphone. When people are really cranking out their data, don't want to forget my phone, then you see, as I said, again, multiple first authors or multiple senior authors, and it's appropriate actually. And there's no difference of whether you're the third last or the last. It's the middle part, right, that can be difficult, and when people are, especially if you have 60 or 70 names. So one of the things is, one of those is to really, during PNDRs, have a letter, for example, from the lead of a study, actually talking about the contribution of that person, and to put that into context that they've decided to work within a team and what their role was in making sure that it got to a certain impact. I think that's important, and so we have to again, probably have a workshop with the dean in which line managers would actually hear that, right? And that's really, really important. I think the other piece for team science is that when you look at the big infrastructure grants that are available, those are all teams, right? Sometimes, though, it's parsed out where it's a bunch of silos altogether, but I don't think that's working anymore. And I think the Biomedical, Research Center is a good example of really trying to pull together team science and work again with this idea of comorbidity and again, a goal alignment around looking at that. And I guess the third thing is that there's a debate always, and it goes back and forth as, what's best? Undifferentiated, stunning basic and discovery research not really aligned to a particular cancer site, or cancer research that's directly aligned to a cancer site and therefore using models and cell lines, et cetera, from that. Both are needed, right? But it's okay to do fundamental research in breast cancer using breast cancer models, et cetera. And so that might come actually from the scientist side of things, and it's okay to do that sort of work. Team science may not be for everybody. That's okay. We all have choices, but it should be an option, a strong option for those that want to be involved. And I would argue that actually it has strong impact, maybe the greatest impact if you can actually carry it out.
Can I just add in, and part of an initiative which is going to be happening over the next year or so, a fund from Research England on research culture, and as part of that, across the university, and Ruth is actually in the audience there, is going to be addressing that exact issue, Simon. How do we roll out team science approach, team research approach maybe, across the university in the way that so successfully has been done through MCRC. So watch this space. Any other questions? If not, I'll take the liberty as chair. Rob, you mentioned about the town halls that you'd had.
Yes.
Are you still doing those? Or because the way you talked about it was we did these and then we moved on to this.
Yeah, well, so we did this in order to force people into a room to try and go for money. And so that was the big carrot at the very, very beginning, coming back to how do we start this? How do you even get people to take time off? Because we had busy clinicians, right, taking time off from their clinics. So actually, it was kind of a fire starter approach when I first came. And, your first 6 to 9 months as a new director, that's your honeymoon period. So you try to do as much as you can and, proceed until apprehended. Yeah, and then I got apprehended, right? And it's like, oh, you're spending this money. And then we had to wait for the return investment, and I think then that was a good thing for the university. So the monies that were apportioned to that were special monies at the time that I had access to as a new incoming director, but they weren't monies that were being regenerated. And then the pandemic hit, and then the Christie and the university weren't working together just because there was financial contracture. Can it happen again going forward? Yeah, absolutely. And I think we are in an era right now where we're talking about the MCRC partners and how we put money back into that innovation space and can do a bit of a free spend around some of these things to really catalyze and pump prime. And that was, from the International Advisory Board. Now these are directors of other, large cancer centers, MD Anderson, even the CEO of American Cancer Society said, for a comprehensive cancer center internationally, you need to have those monies. So we'll see if we get them back. But that's where I would apply to them. As I hinted, there were other ways to do this in the other tumor sites that just came up. CNS, for example, we had the Jeffrey Jefferson come up, and then we had the Brain Tumor Charity Chair. 1.35 million. It's really is a gift, and we'll see how we take it forward. And in head and neck, for example, there was in Radnet, just by happenstance, there was a donor who wanted to give to proton radiotherapy in head and neck cancer. So this comes back to this. Chance favors a prepared mind. And we had just started to have some mini meetings before a full-on town hall. And a donor said, when you go through the Radnet competition, if you talk about head and neck proton and it's well received, we'll give you a donation. So we did that. And so we got $16.5 million for Radnet. And then the donor gave $4.5 million towards head and neck proton radiotherapy for trials and translational studies. Because we showed that we were a strong team and that we could carry out. And actually, that particular gift is actually adjudicated by CRUK. So I have to write about that and show that it's good value for that donor. So I think there were different ways to doing it, but we started off with this very much that, and we wanted to create a buzz. And the last thing I remember in the breast cancer research, there was a patient advocate who kept on looking at me very quizzically when I first did the first 10 minutes, probably because my accent was funny. But part of it was, she really looked, and then she started to partake in things, etc. At the very end of the 2 hours, she came up, she goes, and she was really brusque, she just said, I really thought what you were doing was idiotic. She said, she goes, I just thought it was, I just thought it was OTT, over the top, an over the top American. I said, I'm Canadian, but anyways, over top American telling us all this sort of stuff. But she goes, I really enjoyed that. She goes, I think we did get to something. I haven't seen that happen before. And there was a second project actually about metastatic breast cancer that wasn't chosen, but we actually wound up choosing that 3 years later as part of the Cancer Center. So, positive disruption is okay, and I'm happy to be disrupted in the other way. But I think that was part of it. As I said, the first 6 to 9 months, there's lots of energy coming in and seeing what you could do. I don't think it would have worked in Oxford and Cambridge, but that's all I'm saying, for lots of reasons.
Let's leave it there.
Yep.
Anyway, please,
Thank
let's
you.
thank Rob again.